Today, we're going to tackle a question that is, incredibly personal for so many, but it's also a huge scientific puzzle. And that is, why do so many pregnancies end in early loss? We're going to get right into the microscopic details at the very beginning of life to see if we can find some answers.
Okay, so let's just get right to it. The big question we're exploring today is really simple, but the answer is incredibly complex. Why do some pregnancies end before they've even really had a chance to start? It's a truly heartbreaking thing, and it's something scientists have been trying to figure out for a very, very long time. And now, the answer is finally starting to come into focus.
And it turns out, a massive piece of this whole puzzle is hidden right inside our DNA, in our genetic blueprint. We now know that a major cause of miscarriage and also failed IVF cycles boils down to something called chromosomal abnormalities. Basically, errors in the genetic instruction manual that's supposed to build a person. So, that leads to the next question, right? How in the world do you find these tiny errors? Well, that is where the science comes in. Researchers went on this incredible quest, looking deep inside our cells to try and pinpoint exactly how and why these genetic mistakes are happening in the first place. Now, before we get to the problem, we have to talk about a really key biological process. It's called recombination. The easiest way to think about it is like a genetic shuffle. So, before a parent passes down their chromosomes, the pairs of them actually swap little bits of information. This shuffling is absolutely essential, not just for genetic diversity, but, as we're about to see, for making sure everything gets sorted out correctly. And honestly, this analogy just makes it all click. Imagine a parent's full set of chromosomes is just a deck of cards. Recombination, that process we just talked about, that's the shuffle. The entire goal of this whole biological dance, which is called meiosis, by the way, is to deal out two perfectly even hands. Each hand needs exactly the right number of cards, or chromosomes, for an embryo to have a shot at developing properly.
For the longest time, scientists were really focused on what you might call small errors. Maybe one or two cards getting misdealt. But what recent research found was something way bigger, way more dramatic, a type of error that had pretty much been overlooked.
And this comparison just lays it out so clearly. We've known for a while about single card errors, where maybe just one chromosome gets put in the wrong pile. But what this new research found is what you could call a whole deck error. This is where the entire set of chromosomes fails to separate the right way. It's a totally catastrophic failure in that genetic sorting process.
So here's the absolute kicker. The investigation found that if that initial shuffle, that recombination step, fails completely, it doesn't just mess up one chromosome. Nope, it can throw off the sorting of all the chromosomes at the same time. The entire deal is basically botched from the get-go.
And the researchers also spotted a really clear pattern. You can see it right here on this chart. The frequency of these massive genome-wide errors goes up and up pretty significantly with maternal age. This gave a brand new molecular reason for something that doctors have observed in clinics for decades.
But get this, it gets even more interesting. The study found that these huge errors aren't just random bad luck that happens more as you get older. They actually found evidence that certain individuals have an inherent personal predisposition to this exact all-or-nothing type of mistake.
Okay, so we've just identified a huge fundamental problem in biology. But, understanding a problem is always the very first step toward finding a solution. So now let's talk about how this new knowledge is already helping to crack the case in the real world. So, the big question becomes, if someone is prone to these, let's call them catastrophic misdeals, is there any hope of finding a viable embryo? And the answer, thank goodness, is yes. The goal becomes sorting through all the results of that faulty deal to find that one hand that was actually dealt correctly.
And here's how that works in practice, especially in the world of IVF. First, because of this new research, we can now better identify people who might be at a higher risk. Then, we can use a technology called PGT-A. It's a mouthful, stands for preimplantation genetic testing for aneuploidy. But it basically lets you screen those tiny little embryos at a very early stage. This lets doctors find the ones that got the correct hand, and then they can select those healthy ones for transfer.
At the end of the day, it all comes down to this number, 2. A healthy human embryo needs to be what scientists call diploid. All that means is it has 2 complete sets of chromosomes, one from mom, one from dad. PGT-A is really just a super sophisticated way of counting to make absolutely sure that embryo has exactly 2 sets, not 1, not 3.
So, let's zoom out. What's the big picture here? This research is so much more than just solving a biological mystery. It really reshapes how we think about and approach reproductive medicine and genetic counseling. This table really sums it all up beautifully.
Finding these huge genome-wide errors, that gives a real, concrete explanation for a major cause of reproductive failure. Linking them to age, that helps improve risk assessment. Discovering that there's a predisposed subgroup of people, that allows for way more personalized and effective counseling. And, maybe most importantly, showing that PGT-A can find those viable embryos, well, that offers a direct way to increase IVF success and, hopefully, reduce the pain of miscarriage.
But of course, science is never really done, is it? It's a journey, and this incredible breakthrough opens up a whole host of new questions. Like, what exactly is causing this personal predisposition at a molecular level? Can we come up with a simple test to predict who's at risk before they even start trying for a family? And of course, all these findings need to be confirmed in much larger, more diverse populations of people.
And all of that leaves us with one final, kind of mind-blowing thought. As we get better and better at reading our own genetic code, you have to wonder, could we one day look at a person's genome and predict their entire fertility journey? It is a really powerful question, and it's one that this research brings us just one tantalizing step closer to answering.